Influenza, commonly known as the flu, is an infectious disease caused by an influenza virus. Symptoms can be mild to severe. The most common symptoms include a high fever, runny nose, sore throat, muscle pains, headache, coughing, and feeling tired. These symptoms typically begin 2 days after exposure to the virus and most last less than a week. The cough, however, may last for more than 2 weeks. In children, there may be nausea and vomiting, but these are not common in adults. Nausea and vomiting occur more commonly in the unrelated infection gastroenteritis, which is sometimes inaccurately referred to as stomach flu or 24-hour flu. Complications of influenza may include viral pneumonia, secondary bacterial pneumonia, sinus infections, and worsening of previous health problems such as asthma or heart failure. 3 types of influenza viruses affect people, called Type A, Type B, and Type C. Usually, the virus is spread through the air from coughs or sneezes. This is believed to occur mostly over relatively short distances. It can also be spread by touching surfaces contaminated by the virus and then touching the mouth or eyes. A person may be infectious to others both before and during the time they are sick. The infection may be confirmed by testing the throat, sputum, or nose for the virus. A number of rapid tests are available. However, people may still have the infection if the results are negative. A type of polymerase chain reaction that detects the virus's RNA is more accurate. Frequent hand washing reduces the risk of infection because the virus is inactivated by soap. Wearing a surgical mask is also useful. Yearly vaccinations against influenza are recommended by the World Health Organization for those at high risk. The vaccine is usually effective against 3 or 4 types of influenza. It is usually well tolerated. A vaccine made for 1 year may not be useful in the following year, since the virus evolves rapidly. Antiviral drugs such as the neuraminidase inhibitors oseltamivir among others have been used to treat influenza. Their benefits in those who are otherwise healthy do not appear to be greater than their risks. No benefit has been found in those with other health problems. Influenza spreads around the world in a yearly outbreak, resulting in about 3 to 5 million cases of severe illness and about 250,000 to 500,000 deaths. In the northern and southern parts of the world, outbreaks occur mainly in winter, while in areas around the equator outbreaks may occur at any time of the year. Death occurs mostly in the young, the old, and those with other health problems. Larger outbreaks known as pandemics are less frequent. In the 20th century, 3 influenza pandemics occurred. Spanish influenza in 1918, Asian influenza in 1958, and Hong Kong influenza in 1968, each resulting in more than 1 million deaths. The World Health Organization declared an outbreak of a new type of influenza A, H1N1, to be a pandemic in June 2009. Influenza may also affect other animals, including pigs, horses, and birds. Signs and symptoms. Approximately 33% of people with influenza are asymptomatic. Symptoms of influenza can start quite suddenly 1 to 2 days after infection. Usually the first symptoms are chills or a chilly sensation, but fever is also common early in the infection, with body temperatures ranging from 38 to 39 degrees Celsius. Many people are so ill that they're confined to bed for several days, with aches and pains throughout their bodies, which are worse in their backs and legs. Symptoms of influenza may include fever and extreme coldness, cough, nasal congestion, runny nose, sneezing, body aches, especially joints and throat, fatigue, headache, irritated, watering eyes, reddened eyes, skin, mouth, throat and nose, petechial rash. In children, gastrointestinal symptoms such as diarrhea and abdominal pain. It can be difficult to distinguish between the common cold and influenza in the early stages of these infections. but a flu can be identified by a high fever with a sudden onset and extreme fatigue. Influenza is a mixture of symptoms of common cold and pneumonia, body ache, headache, and fatigue. Diarrhea is not normally a symptom of influenza in adults, although it has been seen in some human cases of the H5N1 bird flu, and can be a symptom in children. The symptoms most reliably seen in influenza are shown in the table to the right. Since antiviral drugs are effective in treating influenza if given early, it can be important to identify cases early. Of the symptoms listed above, the combinations of fever with cough, sore throat or nasal congestion can improve diagnostic accuracy. Two decision analysis studies suggest that during local outbreaks of influenza, the prevalence will be over 70%, and thus patients with any of these combinations of symptoms may be treated with neuraminidase inhibitors without testing. The available laboratory tests for influenza continue to improve. The United States Centers for Disease Control and Prevention maintains an up-to-date summary of available laboratory tests. According to the CDC, rapid diagnostic tests have a sensitivity of 70 to 75% and specificity of 90 to 95% when compared with viral culture. These tests may be especially useful during the influenza season, but in the absence of a local outbreak or peri-influenza season. Occasionally, influenza can cause severe illness including primary viral pneumonia or secondary bacterial pneumonia. The obvious symptom is trouble breathing. In addition, if a child seems to be getting better and then relapses with a high fever, that is a danger sign since this relapse can be bacterial pneumonia. Virology. Types of virus. In virus classification, influenza viruses are RNA viruses that make up 3 of the 5 genera of the family Orthomyxoviridae. Influenza virus A, influenza virus B, influenza virus C. These viruses are only distantly related to the human para-influenza viruses, which are RNA viruses belonging to the paramyxovirus family that are a common cause of respiratory infections in children such as croup, but can also cause a disease similar to influenza in adults. Influenza virus A. This genus has 1 species, Influenza A virus. Wild aquatic birds are the natural hosts for a large variety of influenza A. Occasionally, viruses are transmitted to other species and may then cause devastating outbreaks in domestic poultry or give rise to human influenza pandemics. The type A viruses are the most virulent human pathogens among the 3 influenza types and cause the severest disease. The influenza A virus can be subdivided into different serotypes based on the antibody response to these viruses. The serotypes that have been confirmed in humans, ordered by the number of known human pandemic deaths, are H1N1, which caused Spanish flu in 1918, and swine flu in 2009, H2N2, which caused Asian flu in 1957, H3N2, which caused Hong Kong flu in 1968, H5N1, which caused bird flu in 2004, H7N7, which has unusual zoonotic potential, H1N2, endemic in humans, pigs and birds, H9N2, H7N2, H7N3, H10N7, H7N9. Influenza virus B. This genus has 1 species, Influenza B virus. Influenza B almost exclusively infects humans and is less common than influenza A. The only other animals known to be susceptible to influenza B infection are the seal and the ferret. This type of influenza mutates at a rate 2 to 3 times slower than type A and consequently is less genetically diverse, with only 1 influenza B serotype. As a result of this lack of antigenic diversity, a degree of immunity to influenza B is usually acquired at an early age. However, influenza B mutates enough that lasting immunity is not possible. This reduced rate of antigenic change, combined with its limited host range, ensures that pandemics of influenza B do not occur. Influenza virus C. This genus has one species, influenza C virus, which infects humans, dogs and pigs, sometimes causing both severe illness and local epidemics. However, influenza C is less common than the other types and usually only causes mild disease in children. Structure, properties, and subtype nomenclature. Influenza viruses A, B and C are very similar in overall structure. The virus particle is 80 to 120 nanometers in diameter and usually roughly spherical, although filamentous forms can occur. These filamentous forms are more common in influenza C, which can form cord-like structures up to 500 micrometers long on the surfaces of infected cells. However, despite these varied shapes, the viral particles of all influenza viruses are similar in composition. These are made of a viral envelope containing two main types of glycoproteins, wrapped around a central core. The central core contains the viral RNA genome and other viral proteins that package and protect this RNA. RNA tends to be single-stranded, but in special cases it is double. Unusually for a virus, its genome is not a single piece of nucleic acid. Instead, it contains 7 or 8 pieces of segmented negative sense RNA, each piece of RNA containing either 1 or 2 genes, which code for a gene product. For example, the influenza A genome contains 11 genes on 8 pieces of RNA, encoding for 11 proteins. Hemagglutinin, neuraminidase, nucleoprotein, M1, M2, NS1, NS2, PAR, PB1, PB1F2 and PB2. Hemagglutinin and neuraminidase are the two large glycoproteins on the outside of the viral particles. PAR is a lectin that mediates binding of the virus to target cells and entry of the viral genome into the target cell, while NAR is involved in the release of progeny virus from infected cells, by cleaving sugars that bind the mature viral particles. Thus, these proteins are targets for antiviral drugs. Furthermore, they are antigens to which antibodies can be raised. Influenza A viruses are classified into subtypes based on antibody responses to HA and NA. These different types of HA and NA form the basis of the H and N distinctions in, for example, H5N1. There are 16 H and 9 N subtypes known, but only H1, 2 and 3, and N1 and 2 are commonly found in humans. Replication viruses can replicate only in living cells. Influenza infection and replication is a multi-step process. First, the virus has to bind to and enter the cell, then deliver its genome to a site where it can produce new copies of viral proteins and RNA, assemble these components into new viral particles, and, last, exit the host cell. Influenza viruses bind through hemagglutinin onto sialic acid sugars on the surfaces of epithelial cells, typically in the nose, throat, and lungs of mammals, and intestines of birds. After the hemagglutinin is cleaved by a protease, the cell imports the virus by endocytosis. The intracellular details are still being elucidated. It is known that virions converge to the microtubule organizing center, interact with acidic endosomes and finally enter the target endosomes for genome release. Once inside the cell, the acidic conditions in the endosome cause two events to happen. First part of the hemagglutinin protein fuses the viral envelope with the vacuole's membrane. Then the M2 ion channel allows protons to move through the viral envelope and acidify the core of the virus, which causes the core to disassemble and release the viral RNA and core proteins. The viral RNA molecules, accessory proteins and RNA-dependent RNA polymerase are then released into the cytoplasm. The M2 ion channel is blocked by amantadine drugs, preventing infection. These core proteins and vRNA form a complex that is transported into the cell nucleus, where the RNA-dependent RNA polymerase begins transcribing complementary positive sense vRNA. The vRNA either is exported into the cytoplasm and translated or remains in the nucleus. Newly synthesized viral proteins are either secreted through the Golgi apparatus onto the cell surface or transported back into the nucleus to bind vRNA and form new viral genome particles. Other viral proteins have multiple actions in the host cell, including degrading cellular mRNA and using the released nucleotides for vRNA synthesis, and also inhibiting translation of host cell mRNAs. Negative sense vRNAs that form the genomes of future viruses, RNA-dependent RNA polymerase, and other viral proteins are assembled into a virion. Hemagglutinin and neuraminidase molecules cluster into a bulge in the cell membrane. The vRNA and viral core proteins leave the nucleus and enter this membrane protrusion. The mature virus buds off from the cell in a sphere of host phospholipid membrane, acquiring hemagglutinin and neuraminidase with this membrane coat. As before, the viruses adhere to the cell through hemagglutinin. The mature viruses detach once their neuraminidase has cleaved sialic acid residues from the host cell. After the release of new influenza viruses, the host cell dies. Because of the absence of RNA proofreading enzymes, the RNA-dependent RNA polymerase that copies the viral genome makes an error roughly every 10,000 nucleotides, which is the approximate length of the influenza vRNA. Hence, the majority of newly manufactured influenza viruses are mutants. This causes antigenic drift, which is a slow change in the antigens on the viral surface over time. The separation of the genome into 8 separate segments of vRNA allows mixing or reassortment of vRNAs if more than one type of influenza virus infects a single cell. The resulting rapid change in viral genetics produces antigenic shifts, which are sudden changes from one antigen to another. These sudden large changes allow the virus to infect new host species and quickly overcome protective immunity. This is important in the emergence of pandemics, as discussed below in the section on epidemiology. Mechanism. Transmission in otherwise healthy adults. Influenza virus shedding increases sharply 1 day after infection, peaks on day 2 and persists for an average total duration of 5 days, but can persist as long as 9 days. In those who develop symptoms from experimental infection, symptoms and viral shedding show a similar pattern, but with viral shedding preceding illness by 1 day. Children are much more infectious than adults and shed virus from just before they develop symptoms until 2 weeks after infection. In immunocompromised people, viral shedding can continue for longer than 2 weeks. Influenza can be spread in 3 main ways. By direct transmission, the airborne route, and through hand-to-eye, hand-to-nose, or hand-to-mouth transmission, either from contaminated surfaces or from direct personal contact such as a handshake. The relative importance of these 3 modes of transmission is unclear, and they may all contribute to the spread of the virus. In the airborne route, the droplets that are small enough for people to inhale are 0.5 to 5 micrometers in diameter, and inhaling just 1 droplet might be enough to cause an infection. Although a single sneeze releases up to 40,000 droplets, most of these droplets are quite large and will quickly settle out of the air. How long influenza survives in airborne droplets seems to be influenced by the levels of humidity and UV radiation, with low humidity and a lack of sunlight in winter aiding its survival. As the influenza virus can persist outside of the body, it can also be transmitted by contaminated surfaces such as banknotes, doorknobs, light switches and other household items. The length of time the virus will persist on a surface varies, with the virus surviving for 1 to 2 days on hard, non-porous surfaces such as plastic or metal, for about 15 minutes from dry paper tissues, and only 5 minutes on skin. However, if the virus is present in mucus, this can protect it for longer periods. Avian influenza viruses can survive indefinitely when frozen. They are inactivated by heating to 56 degrees Celsius for a minimum of 60 minutes, as well as by acids. Pathophysiology. The mechanisms by which influenza infection causes symptoms in humans have been studied intensively. One of the mechanisms is believed to be the inhibition of adrenocorticotropic hormone, resulting in lowered cortisol levels. Knowing which genes are carried by a particular strain can help predict how well it will infect humans and how severe this infection will be. For instance, part of the process that allows influenza viruses to invade cells is the cleavage of the viral hemagglutinin protein by any one of several human proteases. In mild and virulent viruses, the structure of the hemagglutinin means that it can only be cleaved by proteases found in the throat and lungs, so these viruses cannot infect other tissues. However, in highly virulent strains, such as H5N1, the hemagglutinin can be cleaved by a wide variety of proteases, allowing the virus to spread throughout the body. The viral hemagglutinin protein is responsible for determining both which species a strain can infect and where in the human respiratory tract a strain of influenza will bind. Strains that are easily transmitted between people have hemagglutinin proteins that bind to receptors in the upper part of the respiratory tract, such as in the nose, throat and mouth. In contrast, the highly lethal H5N1 strain binds to receptors that are mostly found deep in the lungs. This difference in the site of infection may be part of the reason why the H5N1 strain causes severe viral pneumonia in the lungs, but is not easily transmitted by people coughing and sneezing. Common symptoms of the flu such as fever, headaches, and fatigue are the result of the huge amounts of pro-inflammatory cytokines and chemokines produced from influenza-infected cells. In contrast to the rhinovirus that causes the common cold, influenza does cause tissue damage, so symptoms are not entirely due to the inflammatory response. This massive immune response might produce a life-threatening cytokine storm. This effect has been proposed to be the cause of the unusual lethality of both the H5N1 avian influenza and the 1918 pandemic strain. However, another possibility is that these large amounts of cytokines are just a result of the massive levels of viral replication produced by these strains, and the immune response does not itself contribute to the disease.